Hello students, today we are going to start a new chapter, Human Reproduction. In this session, we will discuss about male reproductive organs and spermatogenesis. What are the characteristics of humans? Human beings are sexually reproducing organisms. They are unisexual and they show sexual dimorphism. Sexual dimorphism means they look different physically due to different sex organs. Human beings are viviparous. Let's have a glance of reproductive events in humans. The first event is the formation of gametes, which is known as gametogenesis. The male gamete is called as sperm and its formation is known as spermatogenesis. The female gamete is called as ovum and its formation is known as oogenesis. The second event is the transfer of sperms into the female genital tract, which is known as insemination. The third event is the fusion of male and female gametes, leading to the formation of a zygote. The process is known as fertilization. The zygote divide and redivide and form blastocyst, and it gets attached to the uterine wall. This process is known as implantation. The development of embryo takes place inside the uterus. Once the development of embryo is completed, the mother delivers the baby. There are remarkable differences between the reproductive events in the male and in the female. For example, sperm formation continues even in old men, but formation of ovum ceases in women around the age of 50 years. Male reproductive system consists of a pair of testes, accessory ducts, glands, and male external genitalia. The testes are situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of the testis necessary for spermatogenesis. The sperm formation requires 2 to 2.5 degrees Celsius lower than the normal internal body temperature. In adults, each testis is oval in shape with a length of about 4 to 5 centimeter and a width of about 2 to 3 centimeter. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules. Each testicular lobule contains 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by two types of cells called male germ cells or spermatogonia and Sertoli cells. The male germ cells undergo meiotic divisions, finally leading to the formation of sperms, while Sertoli cells provide nutrition to the germ cells. The regions outside the seminiferous tubules, called interstitial spaces, contain small blood vessels and interstitial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgens. Brachiotestis vas efferentia, epididymis, and vas deferens are the male sex accessory ducts. The seminiferous tubules of the testis open into vas efferentia, through retestis. The vas efferentia leave the testis and open into epididymis located along the posterior surface of each testis. The epididymis leads to the vas deferens that ascends to the abdomen and loops over the urinary bladder. It receives a duct from the seminal vesicle and opens into urethra as the ejaculatory duct. These ducts store and transport the sperm from the testis to the outside through urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called urethral meatus. The penis is the male external genitalia. It is made up of special tissue that helps in erection of the penis to facilitate insemination. The enlarged end of penis, called the glans penis, is covered by a loose fold of skin called foreskin. The male accessory glands include paired seminal vesicles, a prostrate gland, and a paired bulbourethral gland. Secretions of these glands constitute the seminal plasma, which is rich in fructose, calcium, and certain enzymes. The secretions of bulbourethral glands also helps in the lubrication of the penis. Let's see how gametogenesis takes place in males. The male gametes are called as sperms. The formation of sperms can be called as spermatogenesis. It takes place in primary sex organs, that is, the testis. In testis, immature male germ cells, called as spermatogonia, produce sperms by spermatogenesis that begins at puberty. The spermatogonia, present on the inside wall of seminiferous tubules, multiply by mitotic division and increase in numbers. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. A primary spermatocyte completes the first meiotic division, leading to the formation of two equal haploid cells called secondary spermatocytes, which have only 23 chromosomes each. The secondary spermatocytes undergo the second meiotic division to produce four equal haploid spermatids with 23 chromosomes. The spermatids are transformed into spermatozoa or sperms by the process called spermiogenesis. After spermiogenesis, the sperm heads become embedded in the Sertoli cells and are finally released from the seminiferous tubules by the process called spermiation. Various hormones play a very important role in spermatogenesis. At the age of puberty, there is a significant increase in the secretion of gonadotropin releasing hormone, which is a hypothalamic hormone. The increased level of this hormone then acts at the anterior pituitary gland and stimulates secretion of two gonadotropins, luteinizing hormone and follicle stimulating hormone. Luteinizing hormone acts at the Leydig cells and stimulates synthesis and secretion of androgens. Androgens in turn stimulate the process of spermatogenesis. Follicle stimulating hormone acts on the Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. Let's see the structure of a human sperm. It is a microscopic structure composed of a head, neck, a middle piece, and a tail. A plasma membrane envelops the whole body of sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure called as acrosome. The acrosome is filled with enzymes that help fertilization of the ovum. The middle piece possesses numerous mitochondria which produce energy for the movement of tail that facilitates sperm motility essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which, for normal fertility, at least 60% of sperms must have normal shape and size, and for at least 40% of them must show vigorous motility. Sperms released from the seminiferous tubules are transported by accessory ducts. Secretions of epididymis, vas deferens, seminal vesicle, and prostrate are essential for maturation and motility of sperms. The seminal plasma, along with the sperms, constitute the semen. The functions of male accessory ducts and glands are maintained by the testicular hormones called as androgens. Thank you, students, for listening to the session.